Next speaker is Friedrich-Wilhelm Tiller, who will tell us about new avenues of how to measure hemoglobin in stool.
Herr Brenner, I can't thank Frau Dr. Ma for the invitation, but thank you for the invitation. Back to Feces. I think we had a nice time in the temple of expectations and dreams, but Feces will accompany us in CRC screening for the next coming years. And as we in Germany are in the middle of I must say, a battle which screening test is better, GFOBT or FIT, I start with a very clear-cut statement. FIT performed better than a Guyac-based test, and replacement of GFOBT by FIT is overdue. Please remark the nomenclature. We use FIT, which is used by the World Endoscopy Organization, Stephen Harrowan, and FIT is a fecal immunochemical test. And so not to mix G of EBT with I of EBT sounds as if it's the same thing, and you can mix it up. Alternative approaches, detection of CRC-specific DNR arrays in serum or feces, as well as respiratory tests, which have also been reported, wait for clinical validation and are still far from introduction as routine screening procedures. We are in Germany, and in Germany we have a very developed bureaucracy which says where to go and what we get or not. Short words on fecal immunochemical tests. They are based on immune reaction of antibodies to hemoglobin using immunochemical methods to detect and quantify the HB antibody complex. And there's a widely differing methodology, pre-analytics as well as analytics. And they produce widely different analytical results. And please, I stress analytical results. That has nothing to do with a good clinical performance. We talk about analytics, the amount of HB defining an indication for endoscopy. And we have more than 40 FITs in the world market. And finally, we don't know the exact amount of HB which defines a clinical decision, and that's not very satisfying. Why are immunoassays so different? There are lots and many factors. And many of them have been addressed by Stephen Harrowan's Fit for Screening group of the World Endoscopy Organization. But interestingly, everybody circumvented or shunned dealing with the core event in the test, the immune reaction, where antibodies catch a protein, and antibodies have different specificity, have different species, affinity, and so on. And so we think if we want to get out of this mass of tests and we want to standardize something, we have to start with the core event, with the immune reaction. So just to explain to you why these differences exist, we have a molecule, there are different antibodies, they bind to different epitopes on the HB molecules. Certain epitopes are sensitive to masking by other proteins. Different epitopes are sensitive to or resistant to proteolysis. And this is very important because hemoglobin in different patients is present in different form, individual patterns of intact HB and proteolytic fragments, because you see the way of the hemoglobin down this road, and there are plenty of endogenous proteases, the spectrum of bacterial proteases, and the location of polyps and the time of proteolysis differ. So to show you what may happen and what is not considered during when we deal with the analytical parameters is, you see here a protease cleavage site in a HB subunit, and you have two fragments. If the antibody directed or used in the FIT works as in the left part indicated, that's fine, but if the proteolysis has occurred, then we have no signal. So that explains differences in FIT results. So what should be expected from the developed LC-MS reference method? Interestingly, we go the opposite way. You started with a mass of LC-MS results picking out one protein. When we do the opposite thing, we have one protein, or we have many tests and try to reduce it to one LC-MS signal. Well, the matrix is a problem, as we see. So what do we expect? The reference method should not prone to matrix interferences, should not be prone to interference due to non-human HB, and should quantify human HB in feces at levels of at least 1 microgram per gram. I can skip the other. So we initiated with great encouragement by Dr. Ma. Again, thank you for the invitation. We established the MASTAM project, establishing a physical-chemical reference method of HB measurement at concentration relevant for CRC screening, and this is in brackets, development of a procedure to produce a fecal HB reference standard useful for calibration of fit on the basis of a reference method. I said that's in brackets because we are very busy implementing the first step. If there are manufacturers in the audience, they will smile because they did it years ago when they started designing their sophisticated and really very, very highly qualified immunoassays. If you have to deal with the matrix, you have to extract the proteome, you have to find an internal, you have to pre-clean the extract, you have to enrich hemoglobin and to digest, and finally you measure. Extraction and all that is a matter of handling shit, and so nobody loves it, and I agree with Dr. Ma, we would be happy to leave it, but I don't think so. So these are data just showing you to illustrate what we did. We looked that we didn't lose amounts of HB during our procedure, that we had the right yield of proteins after in our procedure, and I would like to focus on the 2 core events in our reference method, that is the LC-SMS. I won't go into details, but I'm absolutely convinced that the laboratory of the future, what you will see is in sequences and a great array of LC-SMS machines that will be and that will come. So I'm happy to have heard about LC-SMS. So what we do is the protein identification by peptide mass fingerprinting. That's, you have a protein and you digest it in peptides and you get an acquired MS spectrum. What we develop, we seek proteins in a database, and we have a predicted MS spectrum having identified these peptides. So this should be a very specific and highly sensitive method approach. The second approach we thought would be very important regarding the matrix feces is introduce an internal standard in the test. That is, at the same time, when we isolate a human HB, we isolate another HB, and it should not be in the human food chain, which had so wonderfully been illustrated by Professor von Zurhausen. So he didn't mention mouse, so we used mouse hemoglobin as an internal standard to verify the extraction process. You see, mouse hemoglobin behaves very, very similar to human hemoglobin. And then we looked in the data bank and we found peptides. This is quite confusing, just believe it, that we found peptides that are different and that can be used as an internal standard mouse hemoglobin and human hemoglobin. So these are the first pictures showing the human peptides when we identified them. This is the extract we used. We did not spike, and that's a question for it. We could not spike native feces and then work on that. It's another question. And here we have the elution times and differentiation of mouse and human chains. So we clearly can differentiate that in our mass spec. So then we look in the samples whether we get the mouse HB at the same time at different concentrations of human HB. And we have a wonderful linearity which is used, which is normal in MS in mass spec for measurements of human HB. So we ventured into native samples. And if you try to investigate a native sample, you have to have it really fresh, and you have to do it in parallel. You can't take a sample from anywhere and then calibrate your reference method. But we had a Ringversuchsprobe. It's the external quality control in Germany, and That looks very nice. It looks as it should look, a fit positive and a fit negative, and that's the blank. And then we had the first clinical samples delivered by Professor Brenner, thank you very much, which we analyzed. We don't know the initial concentrations which you got in your fit studies, but it doesn't matter. I think we have clear-cut signals, and this for one peptide, which is just exemplified in these slides. This is not very much, but it's proof of principle, and to prove the usefulness of the reference method, we should investigate in parallel some fits with our reference method, so we would be able to calibrate the fits in the market to our mass spectrometry, and you would end up in a uniform, and that would be the gist of our study. You would end up in a uniform cutoff value, microgram HB per gram, for all the fits that would undergo calibration with the mass spec method. Standardization is always compromising. So you need to compromise, and you need to agree upon a basis of standardization. This is not nothing is absolute in medicine, and even mass spec does not exclude that there must be some, or can be some HB, but we think it's a useful compromise. What would that mean? That would mean that the uniform cutoff value could be adjusted according to new developments of clinical criteria. If you change your criteria, you have to talk to all the manufacturers, and you have to perform new studies to adjust it. To new developments in diagnostic procedures. I'll show then an example. And to specific criteria of health economics. The number of positive fits determines the number of ensuing endoscopies. So I think not every country is in the wonderful situation of our German ambience, that we have so many endoscopists, I think. So that's another aspect. So let's work out the diagnostic. You all know this last work of Imperiale. He combines a DNA array in feces with 100 nanogram of hemoglobin per milliliter. So what is it? It's a fit. And I think that in future we will have more tests that combine existing tests with new tests. It's never that you get a replacement of an old test by a new one. So how can we cope with that situation if that new tests come, and shall we combine with existing tests? So these tests are knocking on the doors of German healthcare system, and it's quite a lot. Some will be excluded. So selecting fit, and that will apply to every screening procedure, including proteomics, I think. What do we think of first? We think of practicability and economy and price. That's the situation we had. Fortunately, we have now a situation in Germany that clinical performance is evaluated. And I think Professor Brenner will show us then his elegant approach how to validate clinical performance according to sensitivity, specificity, NPV, and PPV in detecting CRC. But we think that this clinical performance should be complemented by analytical criteria, that is the reliability of detecting HB in feces, that is establishing a reference method, and calibrating our fits. So, analytical standardization of fit is desirable, is feasible, provided, and that's the important point, a joint effort of clinicians, laboratory specialists, and manufacturers will be launched. Thank you. I hope I was in time. And that's the group. The group is non-commercial. It's no public funds, no commercial funds, no taxpayers' money wasted. This is Ingrid Frank, immune biologist of our lab. This is Valerie Bochkov of biochemist in Vienna. Wolfgang Bicker added the know-how in healthy MSMS is a forensic toxicologist. Robert May now established the reference lab, which we have at disposal at the Spranger Laboratories in Ingolstadt. That's myself. And Professor Dietrich Seidel is just Hall of Fame, I must say. So, thank you very much.
Thank you very much for this nice overview of important steps towards calibration of fits. Are there any questions from the audience? Yes, please.
Arber from Tel Aviv. Obviously, it's a nice talk. I just want to mention that it's not that easy. And when you're talking about reference lab, etc., it also depends on the time in the year. For instance, in the summer, it has to be kept on cooling. No, well, I'm coming from Israel, so it's all year round. But even here in Germany, I mean, this day, if you take a sample, it might not. And there is also the issue of sending stool by mail.
Yeah, I tell you something. We are in Germany, and we have. In Heidelberg, we have the biggest European commercial lab that goes with cars whenever you want to deliver a sample. So we have a very, very dense system of commercial lab that could manage that. And I would like to make a comment to the question of compliance. If the doctor that administers or that gives the fit to a patient is economically, if he gets something doing the test, you will have a much higher compliance rate. We do not think that it would take longer than, say, one overnight transport in Germany, and the most fits will fulfill this requirement.
So you have the capabilities of doing it overnight?
Absolutely, from morning to the afternoon. Absolutely, and there will be 2 commercial labs knocking at your door. That's the situation. I was running some of these factories for a time,
Yes, please.
Thank you very much. A nice talk. I wasn't expecting so many mass spec talks, and it's nice to hear another one. And so the idea is to use Tandem-Mass-Spec as a reference method for the various fit methods so that they all are playing on a common scale.
We would like to calibrate the various fits.
But because mass spec is then effectively the standard for putting them all together, I'm curious, have you looked to see whether or not when you do the mass spec assay for hemoglobin, does it outperform the fit tests measuring the same analyte with a different technology because it's more precise and is not subject to the interferences?
We just did the first steps. That really has to come. We have to compare fit results with mass spec. That's really still open for discussion.
Right, because mass spec, I'm not a hardware person, but
so am I. Yeah. I would like to have Valery Bochkov here who would tell us that.
But it just occurred to me as you were talking, why do the ELISA? If it's a question of throughput and cost, mass spec is actually getting quite competitive compared to ELISA, and you might have the data for how well it performs. So it'd be interesting to see if you actually develop that.
I hope it won't take too long to present data.
I think this is the issue of clinical validation. Then to see. Maybe the fits would perform better than the gold standard than one would treat.
At least in the field. That's not a routine method. But you never know what mass spec will become out of.
Okay, no more questions. So it's my particular